A very good evening, everyone. A warm welcome to the online platform. Today, we have organized the captivating 20th online lecture series organized by Education Committee of Physiological Society of India. Physiological Society of India is the oldest, renowned, and prestigious educational organization of physiologists. As a part of this organization, I am feeling honored. This organization is doing its commendable job in research areas. The main objective of this organization is to spread the knowledge by using this online platform. PSI has launched this online lecture series on and from July 13, 2023. From that day, PSI Education Committee is trying to conduct one lecture series on the last Saturday of each month. The main utility of this lecture series is to provide the comprehensive knowledge on the various aspects of physiology which are beneficiary to the graduate, postgraduate, and also MBBS students. So, today we have organized this 20th online lecture series, and the theme of this lecture is the environmental toxicology and its remedy. Here myself, Dr. Mousumi Dotto, Assistant Professor of Department of Physiology of Government General Degree College of at Kharagpur 2, would like to invite our today's eminent speaker Dr. Debarati Roy. Presently, Dr. Roy is serving as a visiting faculty at the Department of Food and Nutrition, Taradevi Harakh Kankaria Jain College, Kolkata. Dr. Roy has completed her PhD from Department of Physiology, Kolkata University. She has completed her MSc from University of Kolkata and BSc from the University of Burdwan. She has cracked joint CSIR UGC test and obtained JRF 60th rank in 2015. She has published different research papers in various renowned and prestigious peer-reviewed journals. She is the recipient of ISCA Best Poster Award of the Indian Science Congress Association. Here, her areas of expertise are environmental toxicology, oxidative stress, intestinal mortality, natural antioxidants, etc. With this brief introduction, it is my pleasure to welcome Dr. Debarati Roy. We look forward to your insights on the today's topic entitled Nature's Remedy to Combat Environmental Toxicology. So, welcome you, Dr. Roy. The platform is yours. Thank you.
Thank you, Dr. Dotto, for your present introduction. Very good evening to everyone. I am Debarati Roy. First, I would like to say that I feel very honored. I am grateful to the Physiological Society of India Educational Committee for inviting me as a speaker in the PSI Online Lecture Series 20th session entitled as Nature's Remedy to Combat Environmental Toxicity. So, every living organism resides within the environment, and we the human beings, also being living organisms, need to be reside and to be surrounded by the environment. So the maintenance of the environmental homeostasis is very much necessary for the maintenance and being of every living organism. And environmental toxicity affects the maintenance of homeostasis in the environment. So environmental toxicity can be defined as the negative impact of a substance on the components of environment, including the population of animals, plants, or microbes in the environment. Environmental toxicity is the domain of toxicology that deals with the fate and effects of environmental contaminants on the living organisms residing within the environment. Environmental toxicology was first introduced by the American scientist Rachel Carser in her 1962 book, Silent Spring. She documented the harmful effects of dichlorodiphenyl trichloroethane or DDT on the environment. After the post World War II period, there was a bloom in the manufacturing of many anthropogenic substances, and nowadays it is known that many of the substances possess potential harmful effects on the environment. DDT was the representative of the harmful ingredients in the production of in the use of agricultural fields. The use of DDT, however, was banned after 20 years of its discovery in the agricultural field. So, environmental toxicants are the toxic agents that might be produced by human activities or through natural processes and are introduced into the environment and causes harmful effects towards the environment, including its living organism, and also towards the human health. The environmental toxicants can be categorized depending upon their sources, and they are categorized as organic contaminants and that comprises of carbon skeleton or benzene ring. The inorganic contaminants, that are the inorganic salts of many metals, and the radioactive contaminants. The organic contaminants primarily include the petroleum hydrocarbons, chlorinated solvents, pesticides, coal tar, pharmaceuticals, and additives of food and cosmetics, whereas the inorganic contaminants comprise of inorganic salts of many metals and heavy metals, along with the synthetic polymers like plastics and plasticizers. Radioactive contaminants might be solid elements like uranium, plutonium, or it might also be present in the gaseous form, for example, in case of radon. The heavy metals are one of the major classes of environmental toxicants, and they include the chromium, copper, zinc, arsenic, cadmium, mercury, and lead. The impact of heavy metals on the environment is created as follows. At first, the heavy metals permeate into the environment, therefore, the contamination of environment with heavy metals occurs, and they bioaccumulate in the soil and crop, and from those things they enter into the food chain. After entering into the food chain, the heavy metals get biomagnified, and finally they cause the contamination to human beings, primarily through oral ingestion routes, and it can also cause damages and enter into human body through dermal absorption. The heavy metals that create toxicity to humans include the arsenic. The source of arsenic exposure might occur from pesticide manufacturing plants, and as well as from pesticides, as pesticide manufacturing requires arsenic. The overexposure symptoms of arsenic poisoning include the headaches, drowsiness, confusion, seizures, and it might also become lethal and cause life-threatening complications. Arsenic has been reported to exert its toxic effects on the nervous system, gastrointestinal system, cardiovascular system, as well as on dermal system. The signs and symptoms of arsenic poisoning are the keratosis of palms and soles, palms of hand and soles of foot, and as well as pigmentation in the many regions of body, including the palms, and finally the Bowen's carcinoma, that is the cancer of epithelial cells. Cadmium is also very toxic to human beings. Its source of exposure occurs from the electroplating plants, the battery storages, and from the vapor lamps. Its overexposure causes fatigue, headache, nausea, vomiting, and abdominal cramps with diarrhea and fever. Cadmium is also toxic towards respiratory system, renal system, cardiovascular system, dermal system, and musculoskeletal system. It also causes sensory impairments. One of the signs of cadmium toxicity is the cyanosis or bluish coloration of skin and mucous membrane, and it causes insufficient oxygen supply to the cells. The next heavy metal toxicant is the mercury. Mercury exposure occurs from mainly from chemical industry, and it causes overexposure symptoms like fatigue, depression, lethargy, headaches, etc. It is toxic towards nervous system and behavioral system. It causes a number of behavioral and neural problems in the mercury toxicity contaminated human beings. It is also toxic towards the respiratory system and renal system. Its effect on gastrointestinal system, dermal system, sensory inputs, and other effects like gum inflammation has also been reported. One of the signs of initial mercury poisoning is the erythema of pump, where pink colored dots appear in the open skins like palm and toes and other surfaces of the body. Lead is another heavy metal, very toxic to human beings. Its exposure can occur from the lead production industries and the battery plants, as well as from the workers who are associated with welding and soldering of metal alloys. Overexposure symptoms include the headaches, vomiting, abdominal pain, slurred speech, kidney function alteration, hyperproteinemia, and usually the pale skin color and anemia. It is very toxic towards the nervous system, and it also causes behavioral impairments like incoordination, irritability, depression, and anxiety, and also causes insomnia. It is very toxic towards musculoskeletal system, as it is stored in the bone, and from there it can be released and can cause damage to different organ systems. It is also toxic towards cardiovascular system, reproductive system, and renal system. So, at the cellular level, the heavy metal toxicity occurs by some cellular phenomena. One of them is the substitution of essential ions. Many of the heavy metals are bivalent cations. The major bivalent cation involved in many cellular processes within the body is the calcium. So, heavy metal bivalent cations can substitute the calcium ion, which acts as the second messenger. Substitution of calcium ion can cause many signal transduction defects in many cell types of the body of any organism. Heavy metals are also capable of impairing cell membrane functions like the membrane permeability and other cell membrane functions. It can also interfere with phosphate ions. Phosphate ion is very much essential for production of ATP. So, imbalance in phosphate ion concentration can interfere with energy production in the body. Heavy metals are also capable of imbalancing the homeostasis of sulfhydryl groups. Sulfhydryl groups are very essential redox balance agents. So their impairment in the redox imbalance, so impairment in the sulfhydryl group concentration might cause redox imbalance in this within the cell. And finally, heavy metals are very much competitive in nature for binding sites in the with vital metabolites within a cell. But nature has provided the protection against heavy metal toxicity in the form of phytochemicals. Phytochemicals are a large number of small molecules and secondary metabolites of plants that are very diverse in nature and are categorized into many biosynthetic classes regarding their origin and structure. Heavy phytochemicals include mainly tannins, saponins, alkaloids, and flavonoids. They are capable of chelating heavy metals, and they are capable of reducing heavy metal toxicity as they bind with heavy metal ions and prevent their absorption into the body of the organism. Therefore, they also facilitate the excretion of heavy metals, and as by this means, they mitigate the damage caused by the heavy metals in any organ or tissue. This chelation technique is very effective in the plants that grow naturally on the soil that is affected by heavy metal toxicity. These plants are capable of thriving in the high heavy metal content soil due to their ability to chelate heavy metals. So, heavy metals are seen to confer their protective action against cadmium, mercury, and lead induced hepatotoxicity. As for example, in garlic, allicin is present, and allicin is very effective in this heavy metal induced hepatotoxicity. Garlic extract possesses free radical scavenging activity. It is also anti inflammatory in nature. Its anti cholesterolemic activity reduces cholesterol concentration. It also shows anti-gastric ulcer activity, and it is microbial in nature, antimicrobial in nature. It possesses anti-cancer activity and antioxidant properties. Garlic is also capable of modulating a number of metabolic enzymes within the body. The hepatotoxicity produced by arsenic, cadmium, chromium, copper, lead, and mercury can be protected by the curcumin present in turmeric. Curcumin exerts its protective effect by giving protection in the against histological injury caused by heavy metals. Curcumin is also effective in reducing lipid peroxidation and restoring the glutathione balance, and finally restoring the redox balance of the cell. Many liver antioxidant enzyme activities are restored by curcumin treatment, and curcumin is also effective in restoring mitochondrial dysfunctions exerted by heavy metals. Cadmium induced toxicity can be protected by sulforaphane present in broccoli. And in one experiment, it has been shown that sulforaphane causes protection of human mesenchymal stem cells against cadmium toxicity. So the basic mechanism of this protection conferred by phytochemicals resides in the chelation technique, where phytochemicals, including the flavonoids, tannins, phenolic acid, saponins, and alkaloids, form stable complexes with heavy metal ions and bind them effectively and trap them, finally preventing their effect on the cellular system. Allicin in garlic prevents in this manner the lead and mercury toxicity. The polyacetylens present in cilantro also shows the chelating activity against heavy metals. The curcumin present in turmeric possesses strong antioxidant activity besides the chelating ability, and the catechins present in green tea are effective in binding heavy metals, whereas sulforaphane in broccoli is a potent antioxidant and chelator agent. The next class of environmental toxicant includes pesticides. Pesticides are group of chemicals or agglomeration that are widely used in the agricultural field and farming industry for controlling, annihilating, or fighting off infections or nuisance from the plant and protect humans from disease vectors. Ideally, it was thought that pesticides are very effective against a particular target organism, but in a real scenario it has been seen that pesticides are not target specific, and they can harm human beings also, or other beneficial or any organism besides their target pest. The classes of pesticides include the fumigants, herbicides, fungicides, rodenticides, insecticides, and plant growth regulators. The pesticides, depending on their chemical structure, can be divided into four categories: the organochlorine pesticides, the organophosphorus pesticides, carbamate pesticides, and other classes of chemical pesticides. The organochlorine pesticides contain chlorinated hydrocarbons in their backbone structure. The primary examples of organochlorine pesticides include the DDT, chlordane, aldrin, endrin, linden, etc. The potent toxic effect exerted by organochlorine pesticides can be seen in the nervous system. They are capable of interrupting the nerve impulse transmission. They do this function by inhibiting the sodium channels, the potassium gates, and also by hampering the function of calcium during neurotransmitter release and its secretion. And the more toxic effect is that the organochlorine pesticides are capable of being accumulated as they dissolve very easily in the adipose tissue of any living beings. Therefore, their chance of getting biomagnified is very high. The organophosphorus groups of pesticides are the phosphoric acid esters or thiophosphoric acid esters. They include the renownedly known pesticides like parathion, malathion, diazinon, etc. Their adverse biological effects include interference of hormonal potential. They are also involved in insulin production decrease, and they are capable of disturbing the cellular metabolism of carbohydrate, protein, and lipid. And they also hamper the activities of mitochondria. They generate oxidative stress, and they are capable of interrupting endocrine and nervous functions. One major characteristic feature of organophosphorus pesticides is their ability to inhibit the cholinesterase enzyme functioning, including the inhibition of acetylcholine esterase enzyme. Acetylcholine esterase is very important for neural transmission and transmission of neurotransmitter acetylcholine in the neuromuscular junction for muscle contraction. Inhibition of this enzyme causes hyperactivity in the acetyl choline receptor channels, and they cause entry of cations in unregulated manner. Carbamate pesticides are the N-methyl esters of carbamic acid. Like organophosphate pesticides, carbamates are also involved in inhibiting the activity of acetylcholine esterase. They include carbaryl, ziram, aldicarb, etc. There are other biological process interfering activities include the hormone disruption potential, where they interact with hormone production, and they also cause reproductive effects like male and female reproductive toxicity, and they interfere mitochondrial functions as well as cellular metabolism processes. The main representative carbaryl is heavily used in the agricultural fields. It is used in the dust form, and it has the capability to be hydrolyzed rapidly. The other classes of chemical pesticides include the botanical insecticides, primarily comprising of nicotine. Nicotine is a plant alkaloid found in different plant species, and pyrethrin, which is extracted from chrysanthemum. Similar like pyrethrin, synthetic pyrethrin derivatives called pyrethroids are developed, which has been reported to exhibit hormone interference activity, and they are also capable of influencing reproductive variables in experimental animals. The triazine group of chemical pesticides include the atrazine, which is reported to cause oxidative stress and endocrine activity disruption, cell toxicity, and dopaminergic effect. The nicotinoid pesticides include the thiacloprid, and it is a blocker of it is an inducer of aromatase enzyme activity involved in breast cancer production and in influencing the carcinogenesis in organisms. So, use of these insecticides is very much harmful towards both the environment and also towards human beings. Therefore, some natural agents has been searched, which might be used as natural insecticides. Flavonoids are reported to have natural insecticide activity, as they are capable of deterring attacks from some plant feeding insects. Flavonoids and flavonoid glycosides can be effectively used to manage pest without negatively affecting the beneficial organisms. Therefore, flavonoid and flavonoid glycosides can be used as potential attractants that attracts insects, stimulants, repellents that repels insects, deterrents, and less toxic insecticide uh, agent against the pest, especially true bugs and true flies, and it will not cause any harm toward beneficial organisms. So, flavonoid based insecticides could serve as environmentally benign alternatives to broad spectrum insecticides against some pests, but their field testing is necessary. But if they qualify the field test, they will be effective in conferring the insecticide activity without the toxic effects that are normally produced by synthetic insecticides. Flavonoids as pesticides, as flavonoids are found everywhere in nature. A rich source of flavonoid can be found in our everyday foods, including the berries, the capsicum, the fruits, and the vegetables. Flavonoids are secondary metabolites of plant from both the crop and non-crop varieties, and they are also present in the foods, seeds, and pollen parts. There are more than 4,500 distinct compounds of flavonoids, and they can exist in the plant tissue either in the free form, or they might be conjugated with some sugar molecules forming the glycosides of flavonoids. Flavonoids can be classified chemically into total 6 groups. The two alcohol groups is the flavonols and flavanols. The ketone groups are the flavones, flavonones, isoflavones, and then the anthocyanins. Flavonols and flavones are abundantly found in the foliage of plants, and they create the color diversity in the leaves of plants. Flavonoids as insecticides can act as insecticide in the sawflies. Sawflies damage a large number of crops in agricultural fields. So it has been reported that high concentration of flavonoids, including acacetin, catechin, camphorite, and the flavonoid glycoside myriacetin 3 galactoside, causes harmful effects in the sawflies. There are numerous species of beetles. Some are herbivorous and some are beneficial beetles. The herbivorous beetles are the pests in the agricultural field. In the laboratory tests, it has been shown that the flavonoids, quercetin, taxifolin, pinosembrin, reduce the survival rate in herbivorous beetle, whereas the flavonoids, quercetin, naringenin, catechin, and pinosembrin reduced the feeding behavior in the herbivorous beetle, so that their feeding in the crop or any beneficial plant can be prevented or minimized. Whereas another flavonoid glycoside routine increases the feeding behavior in the herbivorous beetles. So, routine can be used to attract them and for trapping the herbivorous beetles. Another group of pests include the true bugs. They belong from the Heteroptera order, and there are many flavonoid and flavonoid glycosides capable of reducing the survival rate in true bugs, including the quercetin, genistein, and naringenin, whereas the rutin and naringin reduce their survival rate as flavonoid glycosides. Another group of insect pest is the true flies of Diptera order. In the laboratory, it has been shown that high concentration of flavonoids, including genistein, diazein, quercetin, carrageenan, and carangiachromine, reduce the survival rate in the true flies, whereas flavonoid glycosides, naringin, poncerin, and roifolin is also effective in this function. Genistein, diazein, and a nanosuspension of quercetin is very much effective in reducing the survival rates in mosquito larvae. So they can be used to control the larval stage of mosquito. The benefits of using flavonoid and flavonoid glycosides in the pest management occurs as these compounds are toxic to sawflies, herbivorous beetles, true bugs, and true flies, whereas they are not they do not cause any harmful effects in the beneficial insects like butterflies, bees, and predatory beetles. So pest management techniques can be developed by using flavonoid and flavonoid glycosides as insecticides. Flavonoid or flavonoid glycosides can be used as systemic insecticides so that they become incorporated into the plant tissues, and they become naturally resistant towards the attacks of soft fly larvae from their feeding behavior. Plant cultivars can be breeded that express high concentration of flavonoids or flavonoid glycosides so that they naturally become resistant to pesticides, to pests. Genetic modification can be performed to plant species so that they produce high flavonoid concentration and become resistant against herbivory. Flavonoids could also be formulated as an effective feeding deterrent. Rutin, which is a stimulant which causes a stimulation of feeding behavior, can be used as a toxic bait as it increases in the insects pests, as it increases their feeding behavior in high concentrations. And this flavonoids and flavonoid glycosides as insecticides show sublethal effects on growth and can be act as a growth inhibitor, feeding deterrent, or oviposition deterrent so that it impairs with the egg laying capacity of the insects. Formulations can be developed that remain persistent in the aquatic ecosystem as well as in the terrestrial ecosystems to deter insects and pests from the plant. Utilization of nanotechnology with increased research techniques can be implemented to improve the sustainability of flavonoids under field applications in aquatic as well as terrestrial ecosystems. And field experiments are very much necessary to ascertain if the insecticidal activity of these compounds gives protection against pest attacks. One major drawback of using flavonoid and flavonoid glycosides in pest management is that they are required in large quantities for field application. One probable solution might be this, that the agricultural processing waste of citrus orange peels can serve as an abundant and readily available source of flavonoids, and that could be used in mass production of flavonoids using as insecticides. Research are required to improve the persistence of the flavonoid and flavonoid glycosides as the insecticide, as they are used in insecticides. The next category of environmental toxicant includes the polycyclic aromatic hydrocarbons. Polycyclic aromatic hydrocarbons are naturally found in the coal, crude oil, and gasoline, and they are also produced in the air when coal, oil, gas, wood, and tobacco are burned. They are capable of binding with small particles in the air. High temperature cooking and cigarette smoke also contains many forms of polycyclic aromatic hydrocarbons. Polycyclic aromatic hydrocarbons are reported to be teratogenic, carcinogenic, mutagenic, and immunotoxic to living organisms, including humans. Polycyclic aromatic hydrocarbons found in crude oil generally include naphthalene, anthracene, phenanthrene, fluorine, and benzoapyrene, which is very toxic, and they are chemically they are the fused benzene rings of two or more. US Environmental Protection Agency have listed 16 polycyclic aromatic hydrocarbons and are monitoring their frequency and existence in the environment, among which benzoapyrene is very important and very toxic. Oil spill is a phenomena related with the toxicity caused by polycyclic aromatic hydrocarbons. Oil spill occurs due to spilling of oil, mainly the petroleum hydrocarbons. Petroleum hydrocarbons contain a number of polycyclic aromatic hydrocarbons in them as their constituents. Oil spill occurs oil spill can occur in the terrestrial environment as well as in the large ocean surface or any surface of water body. From the terrestrial environment, also the spilled oil ultimately reaches some water body and finally to ocean. In the lakes, rivers, and wetlands, it causes damage of the ecosystem. It can also cause damage to the food chain, including the human food resources. Therefore, oil spills endanger the public health, imperil drinking water, and devastate natural resources, and finally disrupt the economy. Oil in an ocean surface is very presence of oil in ocean surface is very harmful to many forms of life, including the aquatic life, because it prevents the layer of oil prevents the entry of sunlight from environment into the deep water or within water, and it also reduces the level of dissolved oxygen. Crude oil affects the insulating and waterproofing properties of feathers and furs in the body of a number of organisms, including the birds and marine mammals, and they might die from the hypothermia due to this insulation property. Furthermore, ingested oil can be toxic to affected animals and can cause acute toxicity. And the chronic toxicity includes the damage of their habitat and reduction of their reproductive rate. And finally, it can cause decrease in the number of any organism in their population. Oil spills also affects not only the animals but also the plants. Primarily, the mangroves and the saltwater marshes are the victims of oil spill. Oil spill causes the closure of the stomata of leaves and stems. Therefore, the transpiration and respiration processes are affected, and ultimately they are a big threat to the mangrove regions and shoreline and aquatic plants ecosystem. Polycyclic aromatic hydrocarbons are present in oil. Therefore, oil spills are that is present in the ocean surface or the surface of large water body uh, causes evaporation of some semi-volatile organic compounds throughout the time of from weeks to months. This oil that has reached the ocean shore after spending a time in the ocean surface is called the withered oil. The withered oil it reaches the ocean shore and finally creates some semi-solid blobs of crude oil, and these are called the tar balls. Tar balls are recently being seen in the west coast of in India. There are many numbers of tarballs present, and tarball causes polycyclic aromatic hydrocarbon contamination to marine ecosystem as well as shoreline ecosystems. One natural preventive technique can be seen in the forms of tarbol degrading bacteria. There are some microbial organisms capable of degrading tarbols and their constituent polyaromatic hydrocarbons. It has been reported that the four species of bacteria, including the Pseudomonas species strain Betul 14, Pseudomonas species strain Betul M, Alcanivorax species strain Betul O, and Marinobacter species strain Betul 26, together in a consortium are capable of degrading 53 to 97% of N-alkanes and 22 to 61% of polycyclic aromatic hydrocarbons present within the tar balls in just 45 days. So, this consortium of bacteria can provide a solution that minimize the polycyclic aromatic hydrocarbon toxicity present in the tar balls. Another major environmental toxicant is the plastic. Plastics are synthetic polymers. They are completely anthropogenic molecules derived from petrochemicals, means they are created by human activity. Plastics are widely used for their durability, flexibility, versatility, and elasticity. However, their persistent nature in the environment causes long-lasting plastic pollution as they do not biodegrade, and improper disposal and accumulation in the ocean surface harm marine life and disrupt ecosystem and contribute to global environmental challenge of plastic pollution. The consequences of plastic pollution can be seen in the form of the Great Pacific Garbage Patch. The Great Pacific Garbage Patch is an extensive accumulation of plastic debris that circulates with the ocean current and accumulates into large areas of plastic debris islands in the specific regions of ocean. Some plastic islands can be seen in the Pacific Ocean. Here, this is the western garbage patch, and this is the eastern garbage patch seen in the Pacific Ocean. The problem is that not only the intact plastic, but also the tiered plastic called the microplastic is also present in these plastic islands. Another consequence of plastic pollution can be seen in the form of death of numerous numbers of albatross chicks. In the Midway Atoll, albatross chicks are given plastic debris as food, mistakenly thought by their parent bird, and they are given only the plastic debris instead of food. Therefore, they die of ingestion of high amount of plastic and of starving. In the Midway Atoll, a large number of albatross chicks can be found dead with their stomach full of plastic debris. That can be seen in this carcass of albatross chicks. It might cause decrease in the number of albatross population. Another consequence of plastic pollution is the white pollution. White pollution is referred to the environmental pollution caused by plastic. It causes death of huge number of marine as well as terrestrial organisms in each day. Death from plastic entanglement or plastic debris entanglement, and finally from hunger affects a number of organisms and animals in ecosystem. So the mitigating routes can be as follows. The plastic pollution requires sustainable alternatives and sustainable waste management techniques. For this, development of biodegradable polymers can be a part of solution, whereas employing the microbial degradation process can diminish already present plastic pollutants in the environment. implementing eco-friendly substitutes for traditional plastics can be an effective strategy. Technological innovations might help in the waste management strategies in case of plastic. And public awareness campaigns are also necessary so that human beings use less number of plastics and reuse the already present plastic to reduce the plastic using cycle. Policy interventions might be effective to regulate the production and disposal of plastics, and behavioral shift to use less number of plastic is also necessary. Finally, scientific research are required for understanding the environmental impact of plastics and for developing suitable alternatives and solutions so that harmony can exist with the ecological systems. In recent times, it has been shown that a number of microorganisms and fungi species are capable of biodegrading plastic molecules. Aspergillus and Penicillium are the fungi species that contain the enzymes esterase, lipase, and cutinase capable of degrading plastic polymers. This bacteria is the Ideonella sakaiensis. It was found in the waste treatment management plant outside the city of Sakai in Japan. It was found that this bacteria is capable of degrading polyethylene terephthalate plastic, which is the common ingredient used in the manufacturing of water bottles and beverage bottles made up of polycarbonates. And this bacteria use the enzyme PETase, which is responsible for catalyzing the hydrolysis of polyethylene terephthalate into its monomers. Here, these appendages can be seen. This bacteria forms the appendages on the film of polyethylene terephthalate and finally degrades the plastic polymer. Other species of bacteria, including the Bacillus, Proteus, Pseudomonas, and Salmonella, have also been reported to be effective in two types of plastic degradation, that is LDPE or low density polyethylene and HDPE or high density polyethylene. In an experiment, these four bacterial species were isolated from the plastic contaminated soil collected from Valedva Anand, Gujarat of India, and it was seen that 15 to 22% of LDPE plastic and 17 to 24% of HDPE plastics were degraded or biodegraded by the combination of these four bacteria. And in this picture, it is the uninterrupted normal surface of the LDPE plastic, and this surface becomes rough due to microbial degradation of plastic from the biodegrading activity of these bacterial species on plastic film. Another organism that is capable of degrading plastic is called plastivores. Plastivores are plastic eating larvae of some insect species belonging from the order Coleoptera and Lepidoptera. Plastivores consume plastic as for like they eat plastic for and use it for their carbon source, and they are capable of biodegrading plastic polymers including polyethylene, polystyrene, polyvinyl chloride, and polypropylene. Their ability to biodegrade the polymers of plastic is attributed to the microbiota present in their gut. The gut microbiota produces number of enzymes capable of degrading the plastic, but dissociation of the gut microbiota from these insects reduces the plastic degrading activity, indicating that this association of the gut microbiota and the insects of Coleoptera and Lepidoptera larvae is required for better biodegradation of plastic polymers. The plastivores of coleopteran species larvae include the yellow mealworm, dark mealworm, red flower beetle larva, darkling beetle larva, and superworm. whereas the Lepidoptera species larvae include the Indian meal moth, the greater waxworm, and lesser waxworm. Another environmental toxicant agent is the bisphenols. They are the plastic additives and they are used as monomers during polycarbonate plastic production and production of resins. Among many bisphenol compounds, bisphenol A was in use from since many decades, but nowadays its endocrine disrupting capability is causing its use and causing ban in its use in a number of plastic containers, including the plastics used for baby bottle production. Bisphenol compounds are the compounds comprising of two phenol rings, and they are integrated into the polycarbonate based plastic container production, and these containers are used for storage of foodstuffs, beverages, and drinking water. And metal cans that are coated with epoxy resin, that epoxy coat also contains the bisphenol compounds. But due to the ban exerted on bisphenol A, plastic manufacturing industry is shifting towards alternative bisphenol compounds, and bisphenol S is one of the recent alternatives. The problem arises when bisphenol A or bisphenol S or some other bisphenol leaches into the stored foodstuffs from the storage containers. It causes human contamination with the bisphenol compounds, and presence of bisphenol in the human urine confirms the phenomenon that human beings are continuously being exposed to and being contaminated by the bisphenol toxicity. The effect of bisphenol S toxicity on the male reproductive system of rats include the abnormalities of morphology, mobility, viability, and number of epididymal sperms. It also affects the functioning of testicular mitochondria. It induces hypospermatogenesis and changes the Sertoli cells, and it decreases the steroidogenesis due to hampered cholesterol transport to mitochondria. Effect of bisphenol S exposure on the human male reproductive hormone causes decrease in estrogen, serum estrogen concentration, with a decrease in sex hormone binding globulin, and finally a decrease in estrogen is to total testosterone ratio in the serum. Whereas in case of women, polycystic ovarian syndrome is reported to be correlated with urinary bisphenol levels in childbearing aged women. Bisphenol S also produces negative effects on the gastrointestinal system of mammalian small intestine. In the rat, it causes contraction of visceral smooth muscle cells. It increases the acetylcholinesterase activity. It also induces oxidative stress in small intestine. Whereas the bisphenol A toxicity also affects the gastrointestinal system. Bisphenol A causes the decrease in movement of duodenum and alters the nitrogen and alpha adrenergic signaling pathways in small intestine in the experimental rats. Bisphenol A has also been reported to exert toxicity towards liver mitochondria, isolated liver mitochondria, by decreasing the intactness of mitochondria, by inducing oxidative stress and causing oxidative imbalance, and also by decreasing a number of mitochondrial metabolic enzymes like the Krebs cycle enzymes and respiratory chain enzymes. However, one natural agent, gallic acid, has been reported to provide ameliorative effects against bisphenol A toxicity in liver mitochondria. Gallic acid has been reported to protect liver mitochondria against bisphenol A toxicity by restoring the mitochondrial functional and structural properties, by minimizing the harmful effects, negative effects of oxidative stress, and by balancing redox homeostasis, and also by improving mitochondrial metabolism. So the total discussion can be summarized as that there are present a number of anthropogenic and natural environmental toxicants in our environment, and we easily become exposed, we human beings easily become exposed to these environmental toxicants, causing environmental toxicity also in the environment and also within the human body. However, phytochemicals are effective in showing protective action against heavy metal induced toxicity. A number of flavonoids and flavonoid glycosides can be used as substitutes of toxic synthetic insecticide, and polycyclic aromatic hydrocarbon toxicity caused from a number of aerial and water sources can be regulated by a number of bacterial species, whereas plastic pollution, which is a major threat in today's world, can be controlled by biodegradation of plastic through a number of microorganisms and plastivore insects. So finally, it can be concluded that nature has already provided solution for numerous environmental toxicity related problems. However, future research works are very much necessary to discover solutions for already existing environmental toxicity related problems. These are some references of this discussion. And finally, I would like to say that I feel honored to be selected as a speaker on the esteemed PSI Online Series, and towards the organizing committee of Physiological Society of India for providing the opportunity to share the knowledge in this esteemed platform in the form of the topic Nature's Remedy to Combat Environmental Toxicity. And I am also thankful to the respected audience for sharing their valuable time in this discussion period. Thank you.
Thank you so much, Dr. Debarati Roy, for a very, very excellent presentation. I have enjoyed a lot. Actually,
Thank you, ma'am.
myself, Dr. Shutapa Dash, Assistant Professor and Head, Department of Physiology from Rani Indira Devi Government Girls College, Jhargram, West Bengal. On behalf of PSI Education Committee, I extend my sincere thanks to the Almighty for the success of today's program, and it's my privilege to propose a vote of thanks and acknowledge the contribution of those who has worked very much hard work to make this presentation possible. I extend a very hearty vote of thanks to our honorable speaker for today, Dr. Debarati Roy, for presenting very nicely today in this episode of PSI Online Lecture Series. Dr. Roy has nicely discussed in elaboration about the topic entitled Nature's Remedy to Combat Environmental Toxicity. She explained very nicely the natural remedy. So I think this lecture will be very much encourages to the students in future endeavors. The lecture was very much informative as it shows that Dr. Roy has explained very nicely how environmental toxicants affect the various bodily system of human body, and we the especially the humans are how easily get exposed to these environmental toxicants, how arsenic is harmful for the respiratory system, for cardiovascular system, for the nervous system as well as the skin, and on the other way the mercury, how it is very much harmful for our body. She also mentioned the role of pesticides in the agriculture and heavy metals, how disrupts the homeostasis of the body. And phytochemicals like the flavonoids, like saponins, quercetin, etc., how they are acting protective for from these natural toxicants. And she explained the role of pyrethrin in the reproductive system also, where the flavonoids and antioxidants are available in daily life. She also explained it. The polycyclic aromatic hydrocarbons, how much effective for the environment also. She also explained how the oil spills and the plastics are affecting the ecosystem, and she advised for implementing eco-friendly substitutes for avoiding this toxicity. So, according to me, it's a very nice and elaborate presentation, and you have presented also very nicely, Dr. Roy. I'm very much thankful to you. And on behalf of PSI Education Committee, I also again I am giving you the thank. Thank you, Dr. Moshumi Dotto, for since nice coordination of this session. Also, I must mention our deep sense of appreciation to Professor Proshun Priyonayo, President of PSI Education Committee. I would also like to express my sincere thanks to Dr. Kaushik Bharati, Senior Advisor of PSI Education Committee, actor, and Dr. Shompa Sarkar Bishwas, Convener of PSI Education Committee, for covering the whole program in a very excellent way. Thank you all the audience present over here and the all PSI Education Committee members. Thank you all. Good night.